Hi, my name is Soroush Parsa, and I'm an entomologist at CIAT, the International Center for Tropical Agriculture. The overall objective of this video is to demonstrate a method to characterize mechanisms of host plant resistance to herbivores in support of crop breeding efforts.
Our breeding initiative to enhance spittlebug resistance in brachiaria grasses will provide an example. Spittlebugs are serious pests of many economically important grasses, including tropical pastures, sugarcane, and corn. In the American tropics, pastures belonging to the genus Brachiaria are their most important host. A plant can resist herbivore damage through three broad mechanisms: antizoonosis, antibiosis, and tolerance. Antizoonosis, sometimes called non-preference, is the degree to which the plant is avoided when the herbivore is able to select other plants. Antizoonosis is typically detected in choice tests, where herbivores are given access to more than one genotype. Antibiosis is the degree to which the plant affects the fitness of the herbivore feeding on it. Finally, tolerance is the degree to which the plant can withstand or repair damage caused by the herbivore without compromising the herbivore's growth and reproduction. Both antibiosis and tolerance are best detected in no-choice tests, where herbivores are confined and therefore forced to feed on a single plant. Our video will demonstrate a no-choice test designed to estimate antibiosis and tolerance to spittlebug herbivory in brachiaria grasses. Host plant resistance is a key spittlebug management tactic. However, resistance based on strong antibiosis could create selection pressures for more virulent pest biotypes. On the other hand, resistance based on tolerance would allow spittlebug populations to continue